today we are going to start the unit of reproduction that consists of sexual reproduction in flowering plant, human reproduction, and the reproductive health. So today is the first lecture of class 12th, and it is the part of botany, and that is sexual reproduction in flowering plants. So as you guys know, you have studied in class 11th about the morphology in flowering plants, anatomy in flowering plants, and even the plant kingdom, and all the process like photosynthesis in plants and respirations. So these are the lectures that you have studied in the class 11th about the plant kingdom and all those things. And today's in this class we are going to deal about the sexual portion of the flowering plant, that is reproductions. So we are going to start the class, but before that we should know that what is the strategy we are going to use to study this lecture, and what are the in previous years how many questions have been asked from this chapter. We have studied about the morphology in flowering plants, plant kingdom, and reproduction in organism. Okay, so earlier the reproduction in organism was the part of this NCERT, but from after the COVID, that reproduction in organism has been removed from this class 12 chapters. Okay, so sexual reproduction in flowering plants, human reproductions, and reproductive health consist one unit, that is reproductions. So let us start. So as earlier in class 11th, in biological classifications you have studied some characters of the kingdom like Monera, the characters of kingdom Monera, like habitat, structure of Monera, nutrition and reproduction of Monera kingdom, that is bacteria. Even you have studied everything like Protista in fungi, but for plantae you have studied the plant kingdom, the separate chapters, and animalia for the animal kingdoms. So you have already studied about the Monera, Protista and fungi in biological classifications, and for plantae you have studied the plant kingdom, and for animal you have studied the animal kingdom. Okay, so in plant kingdom you have studied about the 5 divisions, that is the lower is algae, then bryophyta, pteridophyta, gymnosperm and angiosperm. But in angiosperm you have studied about the structure like morphology and anatomy. The reproduction part of the angiosperm we are going to deal in class 12 chapter. Okay, so if I ask you what are angiosperm, so what is angiosperm and gymnosperm? When we say gymnos means it's like naked, gymnos means naked, sperma means seed. Like that, angio it's visible and sperma means seed. In simple way, we can say that gymnosperm are those flowering plants which has naked seeds, which we cannot see from our naked eye. And in other way, we can say angiosperms are those flowering plants which we can see from eyes, which the seed we can see, look at it from our eyes. So what are the angiosperms? So there are some features about the angiosperm when we see the flowering plants. So if there is a flowers in the plants, we can say that's our angiosperms. If it is diploid and it has diplontic life cycle and double fertilization, remember it, the double fertilization characters and features of the plants is only found in angiosperm. That is only found in angiosperm, the 7-celled 8-nucleates. So the character like double fertilization, in which consists of triple fusion and the syngamy, that is called referred as double fertilization, that is only present in angiosperm division. Okay, so if you study about any plant, so we studied 2 stages of the plants. The life stages of plants, we found 2 stages. The first one is sporophyte, and the second one is gametophyte. When we say about the gametophyte, so these are the stages of the plants where the formation of gametes takes place, and we know the gamete is a sperm or egg, and it always present in haploid conditions. So gametophyte mitosis takes place, so 2 gametes fuse that fertilize and form the zygote. From the zygote, the development of embryo, the development of seeds, and the development by all the shoots, leaves, plants, roots, everything takes place. That is the part of sporophyte generations. So when we said sporophyte generations, so whether it is leaf, whether it is branches, whether it is roots, everything the ploidy will be diploid, because they all are formed from the sporophytic plant. And when we talk about the gametes, the only gamete is pollen grains, and the egg, that is only gametophyte, and it is haploid. From sporophyte, meiosis takes place, and from diploid, the reduction division takes place and it forms spore, and from spore, the mitosis takes place, and that again mitosis will get into the fusion and form the zygote. So these cycles takes place. These complete the life cycle of the two stages, that is the sporophytic generation and the gametophyte generations. So now we are going to study about the, let us clarify it. What is sporophyte and gametophyte in sperm, angiosperm? So complete plant, that is a sporophyte in angiosperm. So let us try to understand. These are the flowers, and these flowers is diploid. The stem is diploid, the leaf is diploid, even the root is diploid. Why? Because these are sporophytic plants. That is the sporophytic stage of the plant. And if we talk about the gametophyte, so inside stamen and pistil, meiosis occurs to form a spore, and that spore is haploid, and that is the part of gametophyte. So let us consider the another part. So let us see, that is the components of flowers. The pistil is also diploid, the petal, the coloring part of the flower, that is also diploid, the sepal, the green color part of the flower, that is also diploid, the thalamus, the pedicel, everything is that is diploid, and it all arises from the sporophytic part of the plant. So where does gametophyte is present in angiosperm? The question arises. So where we found angiosperm? Where we found gametophyte parts of the plant in angiosperm. So if the plant is a sporophyte, that is diploid, the flower is also diploid. In flower, that microsporangia, in microsporangia, that microspore, that is haploid. Okay, so pistil, that is diploid. The megasporangia, in female part, it forms the ovule. So we found like egg is gametophyte, and the pollen is what? That pollen is also gametes, and that is haploid. We will learn this in later part. So sexual reproduction, what is sexual reproduction? So a structure developed is flower. The structure that is developed in the flower, that is sexual reproduction. So sexual reproduction takes place in the flowers. So before flower developed, morphological and hormonal changes occur. So in any flower, if there will be sexual reproduction takes place, before that there will be some changes in physiology, there is some changes in morphology, and there is some changes happen in hormonal level. And because of that hormonal level physiological changes, the morphology is changed. Okay, so the vegetative bud converts into floral bud. There is essential holes. When we talk about the essential holes, that is androecium and gynoecium, that is essential hole. The corolla, that is petal hole, and the calyx is sepal hole, that is known as accessory hole. So there is two hole, the essential hole, that is the androecium, and gynoecium is essential hole, and the other part like corolla and calyx carries the accessory hole. So now we will start the chapters of sexual reproductions, and we are going to deal about the flowers in all other time. So free fertilization changes. So we are going to divide this lecture in three parts. The first, we are going to deal about the free fertilization changes. After that, we are going to discuss about the fertilization changes, in which we are going to discuss about the double fertilization, syngamy and triple fusion. After that, post fertilization changes, in which we will discuss about the endosperm development, embryo development, and fruit and seed development. So now we will study about the pre-fertilization changes. So before fertilization, what are the changes takes place in the flower, in their morphology, in their physiology? So let us study one by one. So the first one is structure of stamen. So structure of stamen and microsporogenesis. This is the part of androecium, because the stamen, it is the single part, that is androecium is the whole part, and the stamen is the male gametophyte part of the flowers. So let us understand the microsporogenesis and the structure of stamen, that is the part of the male, the structure of pistil and megasporogenesis, that is the part of female, and the third one is we are going to discuss about the other part like pollinations, outbreeding devices. In pollination, we are going to discuss about the self-pollination and the cross-pollinations, and in self-pollinations, we are going to discuss about the autogamy zythnogamy, and in cross pollination we will deal about the xenogamy. After then we will going to discuss about the outbreeding devices like emasculations, bagging, declining, all those things to avoid the self pollinations. So outbreeding devices are the devices to avoid the self pollination and to prevent the inbreeding depressions. So let us start the structure of stamen. So if you have seen the flowers are very sophisticated, and when we see flowers we enjoy, like when we are going to use the flower. So flowers in NCERT, the first paragraph they have written, like how amazed we are. We have seen the different colors of flowers, different varieties of flowers, even in our cultural values, religious values, ornamental purpose, to express our emotions we will use the flowers. There are different types of flowers. There is a different cultural activities. There is a different cultural religion that is based on the flowers. Okay, in different parts of the country we will use, if we go to the South India, the female use the flower and they tie on their back hair. After that we are going to worship the god. When we worship the god we take the flowers. If you are going to express our emotions to the loved ones, we give the flowers. So flowers use even in the happiness, and the flowers even use in the mourning to express our grief. So the flower, so let us study about the structure of flower. So in the structure of flower we are going to discuss about the structure of stamen. So in the structure of stamen, if you have seen, so this one is the part of, so in the stamen we are going to find that the upper part, that is anther, that is bilobed, and the remaining part that is long, that is called filament, and the filament attached to the thalamus or petal. So in 2016 in a NEET they have asked the one questions, and that is about the filament. So let us try to understand. This part is known as, that is connected to the anther. This part that is connected to the anther, that is called distal end, and the remaining part, that is the filament, the basal part of the filament, that is called proximal end, is connected to the thalamus or petal. So in 2016 NEET they have asked about the proximal end. The proximal end of the filament, it attached to what? So the answer is thalamus or petal. And the number of length and the number of filament is variable according to flowers. Different flowers have different number of filament, different number of anther, and different length of filament we found in different types of varieties of the flowers. So that is the anther, the upper part, that bilobe, that is known as anther. So in stamen, two part, the first one is anther and the second one is filament. The filament is long and slender. So now we are going to detailing about the anther. So anther has a typical anther, that is bilobe. So when we cut out, so we are going to found two lobes, and that's why it is known as bilobed. If we cut out from the transverse sections, we are going to found in each lobe there is two theca. In each lobe there is two theca. That's why it is known as dithecous या bithecous. And from the longitudinal grooves we separate two theca. So that's why it is known as dithecous. And from each theca there is a formation of one microsporangia, and that's why if there is four theca it has tetrasporangiate. So sometimes it is also known as tetrasporangiate. So remember, the anther is bilobed, and each lobe there is two theca, and there is two bilobed conditions found. So we can say that is dithecous, and if one bilobe there is two theca, so typical anther is bilobed, each lobe has two theca, and two lobe has four theca, and that four theca has four sporangiate. That's why it is also known as tetrasporangiate. So that one is microsporangia. So look at this diagram. This is the structure of young anther. In young anther we found this one is microsporangia, and in microsporangia it is covered by some layers that is used for the protection of this microsporangia. After the dehydrations, that microsporangia converted after the maturations, that microsporangia converted into the pollen sac and the microspore develop into the pollen grains. So this one is the young anther and that is the mature anther. So in young anther, microsporangia was there, and in mature anther, that microsporangia converted into the pollen sac that contains pollen grains. So this is this diagram of young anther. So we are going to found several layers around the anther. So the outermost layer, that is known as epidermis, the second one is endothermis, the third innermost layer, that is middle layer, and the fourth, the innermost layer, that is tapetum. And from, if you talk about the inner to outer, so the innermost is tapetum, the second outermost is tapetum, after that middle layer, after that endothelium, and after that epidermis. So outermost layer, that is epidermis, and the innermost layer is tapetum. So remember this, they can ask you in from outer to inner in statement wise question. They can ask, please arrange the layers of the anther from outer to inner, or they can ask from inner to outer. Okay, so this one is the sporogenous tissue, and all these things what you are seeing here is all are diploid. These all are diploid. So now we are going to study about the one by one the epidermis. So the epidermis is the outermost and single layer that is used for the protections to the anther. And the second one is endothelium, that is large size cell, and it has radial arrangement, the inner tangential wall. And you know what, that endothelium is used for, that is the part that is used for the pollen dehiscence. See, in NCERT they have mentioned that the epidermis, endothelium and middle layer helps in the protections, and they also help in dehiscence of pollens. But remember, if they will ask you individually that which one is the responsible for the protection, the first one is epidermis, which one is the responsible for the help of dehiscence, that is endothelium. So if they will ask you particularly, they will answer this one, and if they will ask you consist, then all three layers helps in protection and help in dehiscence. So in endotheliums, there is the thickening of cellular. It is cellular thickening. So the cellular thickening, it absorbs water. When they absorb the water, it thickened the structure, and after the thickening, the wall, it helps in dehiscence of pollen grains. And once the water will lose from this cellular thickening, it will dehydrate and the size of the cell will be decreased. And the third layer is, that is middle layer. It is 1 to 3 cell thick, storage and pass food to the tapetum. So let us understand outer three layer role is protection and helping dehiscence, what I said about the NCERT lines. So let us understand, this one is the filament and this one is anther, and the remaining part, that is thalamus. This one is proximal end and this one is distal end. The distal is connected to the anther part, the proximal end is connected to the thalamus part. So now you can see that the whole anther is dependent on one tissue, that is called connective tissues. So now the most important, the fourth one is tapetum. They have asked multiple times in NEET, they have asked multiple times questions from the tapetum. So it one is the most important by the topic and even for the exam purpose. So now understand the innermost layer. The feature is it has dense cytoplasm. Remember this thing, it has dense cytoplasm and it contains bi or multinucleate conditions. What the role of tapetum is to provide nourishment to developing pollens. See, the pollens are haploid. So they have to go through several purposes, like even in pollens there is some vegetative cell and generative cells, so they need food. So for that food and nourishment, the tapetum provide the nourishment to the developing pollen. Even they produce callase enzyme. We will see what is callase, that protect the sporogenous tissues. The sporogenous tissues, before the formation of microspore tetrad, the sporogenous tissues, that what we call megaspore mother cell, microspore mother cell, that microspore mother cell or pollen mother cell, it has some layers sticky around the callose, and to free the microspore tetrad we need to digest that callose. So that's why tapetum produce some callase enzymes that digest the callose and free the microspore tetrad, and it also produce ubis granule which produce exine. produce pollen kit, insect pollinate flower, and tapetum to produce pollen kit. So see, this one is tapetum, and inside we found the sporogenous tissue. Can you tell me the ploidy of a sporogenous tissue? 1, 2, 3. It's diploid, not haploid. So reason why tapetum become bi or multinucleated. So the reason is it has endomitosis, the restitutions nuclei, and polytony. It's not important when you go to the higher classes. You are going to understand about the polytony, but because of endomitosis, that the tapetum nucleus is binucleated or multinucleated. So this one is tapetum. This one is tapetum. So this one is connected with the sporogenous tissues. So sporogenous tissue is connected with some plasmodesmatal connections, and after that, see, even in the sporogenous tissue there is some lining of the callose, and that callose layer is we need to break the callose layer because it is connected. So now each cell is known as pollen mother cell. So the sporogenous tissues, every sporogenous tissue has capacity to produce pollen grains, and even one sporogenous tissue which go to the formation of pollen grains, we can term that is pollen mother cell. So now each cell is known as pollen mother cell. That pollen mother cell undergo meiosis, that is reductional process, and produce 4 microspore. And that microspore, see, around the layer we found the layer of callose, but we have not freed that microspore tetrad. So we need to digest this callose material, and for that digestion tapetum releases the callase enzymes, and that callase enzyme digests callose, and all 4 microspore are freed, and after that mitosis takes place, differentiation takes place, and that microspore converted into the pollen grains. Okay, so from one pollen mother cell it is the formation of 4 microspore, or we can say 4 pollen grains. So now understand the structure of pollen grains. So outermost structure is known as exine, that is produced from the tapetum. The second innermost, that is intine, that is produced from the pollen itself. So and the inner one is cell membrane. So we can say the intine, that is developed from the pollen, that is not the part of gametophyte generation. But here the exine, that is produced from the tapetum, we can say that is the part of sporophytic generations. So now we can compare about the young anther and the mature anther. So let us try to understand. So in young anther we found the epidermis, the outermost, the second innermost endothecium, third middle layer, and the fourth tapetum. When the young anther matured, when the pollen mother cells as sporogenous tissue has converted in the pollen grains, on that time that the middle layer and tapetum are degenerated, or it's consumed. We can say it's consumed. The tapetum, all foods are consumed. Even the middle layer is also used for the storage of food, that's also consumed. So we are going to left only epidermis and endothecium. So in young anther we found that microsporangia, but in mature anther that microsporangia converted into the pollen sac. After that, sporogenous tissue, that was diploid, in which meiosis not occur, but in mature anther the mature meiosis occurs, takes place, and it's converted into the pollen grain, that is male gametophyte. So let us understand the diagram of the NCERT. So we can find the outermost one is epidermis, the second one is endothecium, and the third one is middle layer, and the fourth one is tapetum. And we can find the connective tissue present between the anther, and this one is the sporogenous tissues, that is also diploid. Now understand the second diagram. The outer layer is epidermis, the second one is endothecium, middle layer microspore mother cell, or you can say pollen mother cell, and the tapetum. In tapetum you can see they have mentioned only one nucleus, but it's not like that. It is binucleated or multinucleated. So don't be confused from the diagram. In diagram they have not mentioned about the binucleated multinucleate condition. See, all are single nucleus, but in description they have mentioned about the binucleate or multinucleate conditions. So this one is the mature anther. In mature anther, the outermost layer, the sporogenous tissue converts the pollen grains, the microsporangia converted into the pollen sacs, and only 2 layers, epidermis and endothecium, is present in mature anther. Others are consumed. So see this one, these are pollen sacs. So now we will understand about the microsporogenesis, the structure of pistil, and the microsporogenesis. So in pre-fertilization changes, we have discussed about the structure of stamen, microsporogenesis. Now we will understand about the structure of pistil and megasporogenesis. Okay. So now we will understand about the, we have not discussed about the microsporogenesis. Now we are going to discuss about the microsporogenesis, that is the meiosis. So see, this one is a sporogenous tissue, okay, present in the anther, and it all are diploid condition. The sporogenous tissues in which every cell can say they are able to form the pollen grains. So they will, each cell of the sporogenous can go under meiosis, and they are capable of growing of pollen grains. So these are known as pollen mother cell, or we can say microspore mother cells. So these is the diploid, because these are the part of sporogenous tissues. So sporogenous is also diploid. The pollen mother cell which goes into the meiosis process is also diploid. But after the meiosis, it will form 4 haploid cells, that is known as 4 microspore, and is formed the tetrad, that is called spore tetrad, or we can say microspore tetrad. So after the formation of microspore tetrad, the mitosis takes place. From each microspore, the mitosis takes place, and it forms the pollen grains, in which we found the outer one exine, the inner one entine, the third layer plasma membrane, and in which the large cell is vegetative cell, has irregular nucleus, and in vegetative cell cytoplasm we found the generative cell, and that generative cell is also have dense cytoplasm. So in tapetum we found dense cytoplasm, even in the generative cell we found dense cytoplasm. So now the spore tetrad, the spore tetrad is known as microspore. That microspore divides cytoplasm in unequal conditions and forms the vacuole inside the development of the pollen mother cell, and in that they produce the large vacuole, and because the enlargement of vacuole, the nucleus is pushed away from the center to the periphery region, and after that mitosis takes place. But the spindle formations during the division, it has asymmetric condition, and because of the asymmetric conditions, the division of the cell, it has unequal division, and because of that we found 2 nucleus. The one nucleus has irregular shape and the second one is regular shape. The irregular nucleus has large cell, and the small nucleus has small cell, that is known as generative cell. And why that vegetative cell is large and the generative cell is small? Because of asymmetric spindle formation, and because of that the unequal division takes place. So this one is the generative cell, this one is the generative cell, and this one is the vegetative cell. So let us understand the holistic picture of pollen grains. So that is the mature pollen. The outermost is exine, that is produced by tapetum, and the second one is entine, produced from vegetative cells. The nucleus, which has irregular shape, the plasma membrane, the vegetative cell, that is also haploid, and the generative cell, that is also haploid, developed from the generative cell. And here see, germ pore absent, exine absent, or we can say sporopollenin present. So in pollen grain, the size of the pollen grain ranges from 25 to 50 micrometer, and it is microscopic, male gametophyte and haploid. So now we'll compare the vegetative cell and generative cells. So in vegetative cell we found some similarity. What is the vegetative cell is haploid from single microspore. Even the gametophytic genesis belong developed microspore is also generative cell. So both are genetically same. What is the difference? The vegetative cell has large size, the generative cell has small size. The vegetative cell has large food reserve and the generative cell has food reserve. There is no food reserve. Even in the vegetative cell there is an irregular nucleus and it has cell spindle shape. Even the vegetative cell form the pollen tube and the generative cell produce 2 male gametophyte by mitosis. So now we'll compare about the exine and entine. The exine is made up of sporopollenin, very important. the sporophyte, the exine made up of a sporopollenin, and the sporopollenin is the most resistant organic material which cannot be digested by enzymes, even by alkali, even by temperature, even by acid. So because of this feature of sporopollenin, even it can preserve even in the fossils. So it cannot be digestible. No any organism can digest the sporopollenin. Okay, so it is the most resistant organic matter found in the globe. So now exine, it has sculptured design. We can found different pollen grains, and it has very beautiful, very fascinating architecture outside the pollen grains, the structure of exine. So pollen of one species differ in size, even the design of the exine can be different. So now intine, it is made up of pectin plus cellulose, and it is thin, has continuous layer, and it give pollen tube, and it has a gametophyte nature and is a part of gametophyte generations, and the vegetative cell produce intine. So now the structure of mature pollen. So the structure of mature pollen, it has 2 cell stages, even as 3 cell stages. But in 60% angiosperm, whatever the pollination takes place, it has 2 cell stage, and remaining 40% has 3 cell stage. So what is 2 cell stage? So when pollen is matured, it contains 2 cells. The large one is vegetative cell, the small one is generative cells. So that is known as 2 cell stage. After that, when the generative cells divides, when the generative cells mitosis takes place, the generative cell produces 2 male gametophyte, and that's why it has 3 cell stages, 1 vegetative cell and 2 male gametophyte. Okay, so that is known as 3 cell stage. So when 60% of angiosperm during the pollination, in 60%, 2 cell stages transfer to the stigma, but after the stigma, even they have to convert into the 3 cell stage, and remaining 40% pollination takes place with the 3 cell stage. So give me the answer of the following. What is the ploidy of anther? Tell me, what is the ploidy of anther? That is diploid. What is the ploidy of tapetum? Diploid. Epidermis, diploid. Endothecium, diploid. Pollen mother cell, diploid. Now microspore, haploid. Male gametophyte, haploid. So why anther and pollen mother cell is diploid? Why? Because it generates from the sporophytic generations. So we will stop this class right now, and thank you very much. We'll meet in the next class. Thank you.